Congratulations, everyone. We are nearing the end of aerobic respiration. We'll be wrapping up oxidative phosphorylation by discussing the many ways it's regulated or downright obliterated. The learning goals for today include illustrating the feeder pathways and conditions that control the outputs of aerobic respiration. And then once we do that, we will be assessing the pathophysiology of inborn errors of respiration from case studies and also acquired errors of respiration. Today's molecule of the moment is a class of poisons known as pesticides. These are fish poisons, and I suppose it's kind of like a multiple choice. The question that I'm about to ask you, I'm going to describe two different pesticides to you, and I want you to infer which one is more toxic based off the way it inhibits the electron transfer chain. So let's start with rotenone. Rotenone is derived from the Robinia genus of flowers, and Native Americans used to poison and catch lake fish with this compound. Today, we use it to eliminate invasive fish species, and basically the way it functions is by binding multiple sites within complex 1 of the electron transfer chain, and it's going to inhibit electron transfer of NADH to Q. So we're not going to get as much QH2 generated. And this is the molecular structure of rotenone if you're curious. On the flip side, we have antimycin A. This is another pesticide, and it works a little differently. Instead, it's going to inhibit the oxidation of QH2 to Q at the site of complex 3. So it's going to disrupt the Q cycle, which is everyone's favorite from the electron transfer chain lecture. Both of these compounds are going to be inhibitors of the electron transfer chain, but one of these is more dangerous, rotenone or antimycin A. Use your knowledge of respiration to find the more toxic compound. Looking at cellular respiration at a glance, the primary feeder compounds that fuel the electron transport chain are going to, of course, be NADH and FADH2. So I think a great place to begin is to go through the different stages of respiration that we've covered so far and ask ourselves what are the direct products of these pathways, and ultimately, how many ATP molecules are they going to be worth? So we began this journey with glycolysis, right? And what do we get from splitting one glucose molecule? Well, for starters, we will obtain two ATP molecules. So that's two ATPs in our ATP column, thanks to substrate level phosphorylation. However, we also will be obtaining two NADH molecules from the action of that dehydrogenase. And I want you to remember that glycolysis is occurring in the cytosol, not inside the mitochondria. And that's important because two cytosolic NADHs are ultimately going to be worth either 3 or 5 ATP molecules. And I'm going to explain why there's that variability in a moment, but for now, just accept that all of glycolysis can contribute a total of 5 to 7 ATPs. Now, next up, we do pyruvate oxidation. This is also known as the intermembrane step, and it's going to occur twice per glucose because we get two pyruvates from every glucose molecule. Hence, we're going to get two NADHs when we do these reactions, and it's going to ultimately occur within the mitochondria. So here we can use the PO ratio that we learned last time, where we get 2.5 ATPs for every NADH generated. So 2 times 2.5 equals 5 ATPs made from this step alone per glucose. Oxidizing acetyl-CoA, the product of that intermembrane step, aka the citric acid cycle, is also going to occur twice per glucose, and we are going to get the bulk of our NADH and FADH2 from these reactions. In total, we're generating 6 NADHs per glucose, 2 FADH2s per glucose, and either 2 ATPs or 2 GTPs from that minor instance of substrate level phosphorylation that we observe. So you tally all these up together after multiplying them by their PO ratios, and we're going to obtain a whopping 20 ATPs in that step. Taking it all together, we can anticipate obtaining between 30 or 32 ATP molecules per glucose upon full oxidation. And this is quite the boon because remember, fermentation, anaerobic respiration is only going to give you 2 ATPs per glucose. I now want to talk about how we get the NADH made in the cytosol via glycolysis into the mitochondria, where it's going to be oxidized via the electron transfer chain. And it's not exactly a straightforward process. That being said, I want to maybe calm you down by stating that this process described over here is not nearly as intimidating as it looks, okay? In a nutshell, the inner mitochondrial membrane is rather impermeable to a lot of substrates, one of those being NADH, which in one level makes sense because we want to contain the NADHs that we make via the citric acid cycle and reduce the entropic barrier of unloading them onto the electron transfer chain. So, there's a perk to that for sure, but how do we get the NADHs made via glycolysis into the mitochondria then? Well, we have to get a little bit clever. And the way we do that is through this thing called the malate aspartate shuttle. This is how we ultimately are able to cash in that NADH for ATP. And the way we do that is through two enzymes that are outside the matrix that can convert aspartates into malates. So this enzyme, cytosolic aspartate aminotransferase, will take aspartate, combine it with alpha ketoglutarate, and transfer over an amino group from the aspartate to the alpha ketoglutarate, making oxaloacetate and glutamate as a byproduct. From there, a second enzyme, cytosolic malate dehydrogenase, can reduce oxaloacetates with NADH made via glycolysis and convert it into malates. And all of this is happening within the intermembrane space, mind you, all right, on the front side of the inner membrane, the impermeable one. And this is useful because malate can cross into the matrix, go through the inner membrane, through the activity of this malate alpha ketoglutarate transporter protein. So we're going to antiport one equivalent of alpha ketoglutarate out of the mitochondrial matrix and then sneak into with its place an equivalent of malate. And this is really helpful because this alpha ketoglutarate can go feed into that enzyme process, and malate can be activated then by mitochondrial malate dehydrogenase, which is going to do the opposite reaction. Take malate, back convert it into oxaloacetate, and then, oh look, we're going to have mitochondrial aspartate aminotransferase do the opposite again, back to aspartate. So we're undoing the reactions in the intermembrane space, which means we're going to take an equivalent of NAD plus and then reduce it to NADH, which is free to then drop off its electrons to the electron transfer chain. The aspartate then, of course, can bleed back out through an aspartate glutamate transporter, and then the cycle continues. What are we doing here? We're not physically transporting NADH across that inner membrane, but it's a clever and indirect way to delete an NADH on the outside and then generate an NADH on the inside. Overall, this is a lossless net import of NADH into the matrix. We invest NADH into a molecule over here and then extract it back out over there. In addition to the malate aspartate shuttle, there's also the glycerol 3-phosphate shuttle, which also serves to indirectly transfer reducing power into the mitochondria via this chaperone metabolite. And the chaperone here is going to be generated from dihydroxyacetone phosphates, which is a really prevalent metabolic intermediate. And what we're going to do here is juice it up into the chaperone molecule, glycerol 3-phosphate, through the activity of this cytosolic enzyme called glycerol 3-phosphate dehydrogenase. It is going to take, just like before, NADH made in the cytosol via glycolysis and reduce DHAP into this glycerol derivative. Next, we have a special channel protein specific for glycerol 3-phosphate that pumps it in to the intramembrane space of the mitochondrion. From there, a single enzyme that resembles complex 2 is going to oxidize glycerol 3-phosphate back into dihydroxyacetone phosphate, which we can pump back out into the cytosol, complete the cycle. And oh look, when we oxidize glycerol 3-phosphate, we are in turn going to be reducing FAD into FADH2, which is what complex 2 does. And then that's not all. Complex 2 then transfers the electrons from FADH2 onto coenzyme Q, reducing it into ubiquinol, QH2. And that is free to migrate down the inner mitochondrial membrane and then participate in the reactions of the Q cycle at complex 3. In short, what are we doing here? We have, again, an equivalent of cytosolic NADH entering the electron transfer chain, but it's entering it as an equivalent of FADH2. We're converting NADH to FADH2. This shuttle system is therefore less efficient than the malate aspartate system because we know that NADH has a higher PO ratio than FADH2. Now, why is this the case? Why are we losing out through this inefficient conversion process? Well, on one hand, first of all, biology doesn't care if a process is imperfect. It just cares if it's good enough. And this on its own most certainly is. However, you can look at this from a glass half full perspective. There can be benefits interpreted from this less efficient feeder pathway. For example, one potential perk is tunability. The cell doesn't always want to be chugging out ATP at full throttle. Think of it this way. The malate aspartate system is going to be more prevalent in skeletal muscle, where it benefits this flooring of the gas pedal. The glycerol method is kind of like tapping the gas pedal. It's better for general use. We don't need to go full throttle all the time. Secondly, the benefit of this additional feeder pathway is diversity of access. Consider this: if your complex 1 or complex 2 is down thanks to rotenone, this is going to give you a fighting chance. Bam, we have a back door into aerobic respiration. Mitochondrial glycerol 3-', dehydrogenase resembles complex 2, but it is structurally different from complex 2. If our complexes are transiently inhibited, we're not completely dead in the water here. Let's now discuss hypoxia's effects on aerobic respiration. Recall that we need a terminal electron acceptor like oxygen at the end of the electron transport chain. This is what's going to keep that wireless current alive within that inner mitochondrial membrane, and that's going to keep the proton pumps active and continuing to actively transport protons into the intermembrane space. So said another way, ATP synthesis is only going to occur when we have, well, oxygen as a terminal electron acceptor, of course, and when the concentration of protons in the intermembrane space is exceeding that of the mitochondrial matrix. We need to have that gradient. The pH of the intermembrane space needs to be lower relative to that of the mitochondrial matrix. If we run out of oxygen, that will ultimately impede the function of our proton pumps. So the protons are going to gather in the mitochondrial matrix, which is going to result in a rapid pH drop. And this makes sense. The citric acid cycle to begin with is, big surprise, acidic. There's lots of acids accumulating in that zone. And that's going to be a major problem because ATP synthase technically works in both directions. When the proton concentration is high in the intermembrane space, that is going to induce one kind of rotation of the rotor of ATP synthase in a way that's going to catalyze the formation of ATP. However, when we are hypoxic, that is going to cause protons to accumulate on the matrix side instead. And therefore, diffusion is going to occur in the reverse direction, which is going to reverse the direction in which these gears rotate. When that happens, ATP synthase is going to catalyze the reverse reaction, aka the destruction or hydrolysis of ATP. So we're not only not making ATP in hypoxic conditions, which is bad enough, but we are burning what precious supply remains through this reverse action of ATP synthase. This is a huge, huge problem. It's almost hilarious how bad this is. But thankfully, biology has a hack for this, and it's called IF1. There is a protein called IF1 that combines the gamma paddle of ATP synthase, which is responsible for driving that rotational catalysis. And when the pH drops, it will change the protonation state and cause it to form these salt bridges that will make it dimerize and clamp together like so, and halt two ATP synthases. It's kind of like throwing a wrench into the spokes of a wheel or a gear. It's going to mechanically jam the enzyme. It will inhibit this sadistic reverse ATP synthesis process until we restore the proper directionality of our proton gradients. This is not the only problem hypoxia creates for the mitochondria. Low oxygen conditions is going to ultimately create an electron traffic jam in the ETC. And this is how. Ultimately, the electrons injected into complexes 1 and 2 are going to funnel along and then eventually off-ramp the highway onto an oxygen molecule to form water. If oxygen is low, this process at complex 4 slows down. So we're going to create a backup that's going to trickle up all the way to complex 1. And these electron shuttles that we're generating, QH2, cytochrome C, they're going to have nowhere to go. And these stalled electrons are not going to wait indefinitely on the broken down electron highway. So what they're going to do is they're going to find another place to stick these rather reactive electrons, such as residual oxygen in the vicinity, turning it into superoxide or peroxide, really dangerous radicals. Or we can dump the electrons onto the membrane itself. Many membrane lipids are going to have radical sensitive alkenes in there. And that's going to create this nasty radical propagation, and it's going to destroy the membrane and start infecting all of the useful molecules with this radical cascade. Radicals are dangerous and frankly contagious. We need a way to quench these radicals quickly before they completely get out of hand. One method that you may recall involves aconitase, the enzyme responsible for scrambling up citrate into isocitrate. It's going to contain a very radical sensitive cofactor, the iron sulfur cubane, and any accumulation of free radicals is going to obliterate that essential cofactor and irreversibly inhibit that enzyme. That is going to put some very hard and brutal brakes on the citric acid cycle, and thus the generation of NADH and FADH2. This is going to ease the traffic going onto the clogged highway and ultimately reduce the formation of radicals, preventing this disaster from becoming a catastrophe. There's another strategy that we could employ to attenuate the generation of reactive oxygen species, ROSs, radicals, and that's through the activity of antioxidants. Glutathione is a perhaps gentler way of dealing with maybe trace accumulation of radicals. It's a sulfur-containing compound, and what it's going to do is directly intercept and neutralize reactive oxygen species like hydrogen peroxide, and it's going to destroy the glutathione into a glutathione dimer through the activity of glutathione peroxidase, and it's going to essentially convert the ROS into harmless water. So these are our options for dealing with radicals generated through hypoxia. If it's an acute instance, we can whip out the emergency brake. If it's a little bit of radical, maybe from a transient hypoxic condition, we can tap the brakes. Our good old friend hypoxia-inducible factor is going to make an appearance again, and by now hopefully you can appreciate why it's considered to be this master regulator of oxygen homeostasis. To recap, its activation is going to upregulate glycolysis through many different mechanisms. For starters, it's going to increase transcription for more glucose transporters. This is going to increase the intracellular concentration of glucose. In addition to that, we can upregulate 8 out of the 10 glycolytic enzymes, thus increasing the cell's capacity for glycolytic flux. And both of these are meant to compensate for the decreased capacity to make ATP through aerobic respiration in hypoxic conditions. And we've learned just now that attempting to do aerobic respiration in the absence of oxygen is a recipe for disaster. So, as an additional layer of protection, HIF1 is going to downregulate all of these oxygen-dependent pathways, and this is how. For starters, it's going to increase transcription of pyruvate dehydrogenase kinase. This enzyme is going to inactivate PDH and thus inactivates the intermembrane step, thus shutting down the entire citric acid cycle. Additionally, hypoxia-inducible factor is going to directly impede the electron transfer chain itself by making more of this IF1 protein, that monkey wrench that gets thrown into the ATP synthase complexes when the electrochemical gradient gets reversed. Hypoxia-inducible factor 1 is also going to encode for a protease that will specifically degrade a subunit in complex 4, and we're going to replace it with a more oxygen efficient subunit that's ultimately not going to be as ATP efficient, but it's going to reduce the demand for oxygen and thus protect us a little bit from this reactive oxygen species generation. And that's really the core principle here. All of these changes are going to work together to greatly mitigate the production of radicals. And it's all thanks to this factor. And it achieves that by decreasing the activity of our oxygen-dependent pathways and increasing flux through the anaerobic ones. Two additional very important parameters that regulate the flux of respiration in the cell are going to be the energy charge of the cell, which we've talked about before. You can sort of think of it as the ratio of ATP to ADP, and also the cell's redox state, which is going to be its ratio of NAD plus oxidized to NADH reduced. And these metabolites are going to be highly influential in controlling the rates of the following processes, starting with glycolysis, of course, the intermembrane step occurring right after, which bridges glycolysis to the citric acid cycle, and then ultimately oxidative phosphorylation. And the common theme here is that all of these steps shown in this slide are going to be the choke points, aka the regulatory points of these reactions. By upregulating or downregulating the gatekeepers, that is going to dictate what the overall flux of these reactions are going to be. And so there's really no new information on this slide. I just think it's going to be an excellent study tool for you moving forward. What's fascinating about aerobic respiration is that many organisms break it on purpose. When animals hibernate, for example, they need a way to produce heat throughout the winter. And the way they do that is, first of all, by packing on a lot of weight in the form of brown adipose tissue. You can see in these histological samples that brown adipose is different from white adipose because it has to do with the number of mitochondria present. These large fat droplets don't really have any mitochondria in them that I'm seeing. These are really quiescent tissues, but the adipose tissue in hibernating animals is very active based off the number of these brown mitochondria spotting the slide. People think that winter weight is there primarily to serve as insulation from the cold weather, and that is partly true. But really, it is these mitochondria that will burn these fuel stores in a deliberately wasteful manner that's going to prioritize the production of heat over ATP. The mitochondria shown here in hibernating brown adipose tissue is going to make a very special protein called thermogenin. It is also known as uncoupling protein 1. Thermogenin is a transmembrane channel in the inner membrane of the mitochondrion that lets protons through. It's basically ATP synthase, but without the synthase portion. We are deliberately wasting this proton gradient that our pumps worked so hard to generate. And this sounds like a catastrophe, and normally it would be, but this is actually quite deliberate in hibernating creatures. The electrochemical potential generated by these pumps is a form of potential energy. By bleeding the protons from the intermembrane space back into the mitochondria without converting it into chemical energy, that means we have to convert it into another form of energy, because energy and matter can't be created or destroyed, only transformed. So what kind of energy do we convert the potential energy into? Thermal energy. Uncoupling the proton diffusion from ATP synthesis is going to generate a lot of heat without shivering. And that is what's going to keep the animals warm without waking them throughout the winter months. And sleeping bears can get away with this because they don't need a lot of ATP while they are sleeping, and they have calories to spare thanks to this abundance of adipose tissue. They do, however, need heat though. Now, uncouplers, which are compounds that bleed the protons back into the matrix without making ATP, they aren't always good compounds. Dinitrophenol is a small molecule that also functions as a proton coupler. It's not a channel protein, but this is a rare example of an acidic compound that can freely diffuse across membranes while carrying that proton on this phenol moiety. And it's able to just drag the protons down into the matrix side. Just simple diffusion. And this is a poison that's also going to make the activity of the pumps futile and convert the potential energy of the electrochemical gradients again into heat. Some of the nastiest poisons out there actually target the electron transport chain. And since 95% of glucose's energy is derived from chemiosmosis, if you disturb that, well, you're going to disturb 95% of the cell's ATP output. And the problems, unfortunately, aren't localized to ATP production. Clogging this electron highway, particularly in non-hypoxic conditions, is going to result in a rapid generation of reactive oxygen species, radicals. These are highly dangerous, and we do not want these to be made. Additionally, any blockade upon aerobic respiration is going to prompt the cell to compensate by upregulating glycolysis and lactic acid fermentation, these anaerobic pathways. And that may work as a short-term solution, but you're going to quickly generate lactic acid and result in acidosis, which is also a very big problem. And lastly, if we keep on blocking the electron transfer chain, that's going to cause our NADHs to back up and accumulate since they won't be able to offload their electrons onto complex 1 and beyond. And that's going to cause the malate aspartate shuttle to reverse its equilibrium, and NADH is going to indirectly trickle into the cytosol and mess up the redox state of the cell, which is also going to have a negative effect on glycolysis, which is the only thing left keeping the cell alive. So you can perhaps appreciate how breaking down the ETC really quickly ripples out the problems beyond the walls of the mitochondrion. In this figure, we have marked up the different ETC poisons that you should be broadly aware of. Rotenone, you may recall from our molecule of the moment, and amytol both inhibit complex 1. Antimycin A is also potentially familiar. That's an inhibitor for complex 3. Both of these are going to interfere with Q metabolism. Malonate is a competitive inhibitor for complex 2. So that will also screw up the Q pool, but it's also going to negatively impact the citric acid cycle as well because complex 2 moonlights between these two pathways. Oligomycin is a really sneaky compound. It's going to physically clog the half channels of ATP synthase. So it doesn't matter if we have functional proton pumps, none of them are going through ATP synthase anymore to churn the gears for rotational catalysis, which means no ATP gets made. And lastly, I want to talk about the complex 4 inhibitors, azide. Cyanide, and carbon monoxide. All of these compounds are going to disrupt terminal reduction, the dumping off of electrons onto molecular oxygen at complex 4. And the reason why they do that is because complex 4 relies on a heme cofactor working together with this copper cofactor to bind and reduce dioxygen. And maybe you recall from the protein lectures that carbon monoxide is going to bind heme and hemoglobin really tightly and displace oxygen. And essentially, the same thing is going to happen for all 3 of these poisons. They are going to insert themselves over here, not get reduced, and never leave, making these 3 compounds some of the most vile, painful, and fast-acting poisons out there. Because not only are they messing up the electron transfer chain, chances are they're going to be messing up your hemoglobin as well. Mitochondria are fascinating beasts because, unfortunately, they tend to be hotbeds of genetic mutation. And I mean, think about it. They operate under highly oxidizing environments, and that's bad for DNA. And remember that the mitochondria have their own, and they are going to code for their own respiratory machinery. Not only that, there are several mitochondria inside each cell. So chances are that one of them is mutated and goes awry is sadly non-zero. And that's not necessarily the end of the world, particularly if it's a very large cell, because that would mean that it has many mitochondria and the rest of the functional ones can pick up the slack for the mutated one. And I want you to also think about, well, what is the largest kind of mammalian cell that you are aware of? Chances are it's going to be the ovum or oocytes, egg cells. These are very large, very active, and therefore possess a large number of mitochondria. You may also recall from developmental biology that mitochondria are matrilineal. They are maternally inherited. All of your mitochondria come from your mother's impregnated ovum. And so let's do a little bit of a thought experiment now. Let's pretend that we have a diploid cell, some sort of a progenitor, and maybe 20% of the mitochondria have a defective complex 1, an LHON mutant. That stands for Leber's hereditary ocular neuropathy. And we are going to divide the diploid cell through meiosis into haploid egg cells. And through random chance, some of those bad mitochondria might get concentrated into one of these eggs, and perhaps the healthier mitochondria get randomly sorted into another egg cell. Then we're going to fertilize the egg with a sperm cell, and it's not going to donate any mitochondria into the ovum. And that is going to result in potential embryos with very different mitochondrial compositions. This ovum unfortunately inherited a lot of these mutant mitochondria, 80% of them. So it's possible that the fertilized egg may become non-viable or result in a child with very severe disease. An ovum that has a smaller percentage of inherited mitochondria may result in a child that has relatively few symptoms, or maybe something in between. So the fact that we have this variability is called mitochondrial mosaicism. And mosaicism can get even more complicated when you think about the blastulation process. What happens to an egg cell upon fertilization? I've just drawn an egg over here. I'm going to draw little dashes to represent good mitochondria, and these purple circles are going to represent bad ones, and they just happen to be clustered in that quadrant that I've just drawn. So the cell is going to start rapidly dividing. And if you've noticed, we have concentrated the bad mitochondria into this part of the developing blastula. And if those tissues become something ultimately quiescent, such as eye tissue, which is the case in hereditary ocular neuropathy, it's going to be problematic for the person that this blastula becomes, but perhaps they can live with it and have a somewhat normal quality of life. If those mitochondria that are bad are scattered throughout evenly, well, maybe it won't even have a noticeable phenotype. If those mitochondria get populated into progenitor cells that become, I don't know, the brain or the heart, then that can create severe problems early on. So mitochondrial mosaicism, the possibility to have these different manifestations of these phenotypes, can be a really challenging problem to parse out.